We have opportunities in areas like diagnostics that are potentially significant because what we've been able to show with our stemECHO platform is that we can actually grow up cells from tumor biopsies, and then we can actually run existing drugs against them to see if the drugs work in killing the stem cells. We can also do this in the future with the new therapies that we develop, and we can create biomarkers or assays that we can readily test our new drugs against to understand whether or not they'll work positively in patients. For instance, we've already shown that we can take cells from biopsies and we can actually grow them up within 10 days and test them against drugs that are currently available to understand which drugs to specifically treat that patient tumor with. And the opportunity is significant because they improve outcomes and reduce cost.
So we are pushing forward with this TP-101 drug combination, which is just remarkably effective across a broad range of cancers and an increasingly broad range of chronic inflammatory diseases. That being said, we have found cancers that are resistant to this and continue to develop even triple combinations that augment the response of those cells. So the flexibility of the system is immense. And we have the feeling that we can, in an iterative fashion, figure out how to address more and more of these cancers. The importance of stemECHO's ability to clone cells is that you can get an unlimited number of offspring from that same cell. That allows you to screen against 10,000 or 100,000 different drugs in parallel with the normal clone stem cell, and it becomes very easy to do drug development. You say, find me a drug that kills the bad guy and doesn't hurt the good cells, and you win.
Another big opportunity for us is in regenerative medicine. One of the things that we could potentially do with the stemECHO platform is isolate individual stem cells from organs, diseased organs, isolate out the healthy ones, grow up healthy organ tissue, and replant it back into patients. This eliminates the need for immunosuppressive therapies, which is one of the biggest challenges for patients that are receiving organ transplants today.
We could imagine these patient-derived normal stem cells can be very useful for regenerative medicine, and these patient-specific pathogenic stem cells could be used for the personalized medicine, because using them, we can test whether the drug is really effective to that patient before we give to the patient. And if this can be done, and we need a very automatic system to help to scale up our laboratory, so we can quickly feed back the information to the patient.
Many of these patients who need organ transplantation are on important but very powerful drugs that have their own side effects. As you know, there's a huge shortage of organs to give to patients who need transplantation. And many patients who've had organ transplants eventually reject the organ that they have. And so there's a massive need to find better ways to help these patients who need transplants. And in this field, if you can actually grow normal organs from patients, so-called regenerative science, one day this science could be turned to replacing damaged organs like a damaged lung or damaged pancreas or a damaged colon or heart from a patient, and they wouldn't need to be put on any therapy because they grew it themselves.